Nervous system has many cells other than neurons. In this video, we are going to see what are these cellular components of nervous system and how they influence the functioning of nervous system. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you have subscribed my channel. You can also follow HM Learnings on Facebook, Instagram and Twitter for daily updates. We have also started uploading all our content on website called as SlideShare. You can easily download and clip the slides on that website. I have also started sharing the SlideShare link of each video in the description box of that video. So you just need to click on that link and you will be on the SlideShare. We have also started giving live online classes of physiology in which there will be direct discussion by me. So you can easily ask me anything directly. To know more, you can contact us on this email ID or you can directly message us on our official pages. So with this, we start our today's video, that is the cells of nervous system. So nervous system has two type of cells. It has neuron, or also called as a nerve cell, and the another type of the cell is the glia cell, also called as a neuroglia cell. So I have already finished with the neuron video. In that video, I have discussed about the parts of the neuron, the classification of the neuron also. So I will share the link in description box and in I button. You can go and check that video. So in this video, we will just focus on the other type of the cell present in the nervous system, which is the glia cell. So glia basically means, it is a Greek word which means glue. So it acts as a connective tissue in the nervous system. It actually keeps the neurons together and it has many other functions also, like it secretes the extracellular matrix, it plays metabolic and nutritive roles, it acts as a sink and a source of the ions, it insulates the axons and the synapses by making the myelin sheet around them, and they also phagocytose the neural debris. So what are the types of the glia cells? There are basically two types of the glia cells, macro and the micro. So micro is only one type of the cell, but the macro has other types of the cells also, like the Astrocytes, Oligodendrocytes, Swan cells and the Ependyma cells. So one by one we are going to see their function, their structure, and then we will talk that how they influence the functioning of the nervous system. Okay, so first we talk about, before that we need to talk about the difference between neuron and the neuroglia cell. What is the difference and how they are different on their functional aspect. Like neuron has a function in conducting the impulse. It basically conducts the impulse in the form of action potential. But neuroglia cell is not that like cell. It is not conducting any impulse. Neurons will form the synapse with the another neuron, but the neuroglia cell doesn't form any type of the synapse. Neuron has a communicating function. It communicates, it transfers the impulse from one part of the body to the another part of the body. But the neuroglia cell has the supporting function in the nervous system. It supports the neurons. Neuron, we know that it is a highly differentiated cell. It actually lost their ability to divide. But in the case of neuroglia cells, they still have the ability to divide. Okay, then so the first type of the macroglia cell is the oligodendrocyte. So oligodendrocyte is actually present in the central nervous system and it forms the myelin sheet in the central nervous system. So you can see in this diagram that this is the oligodendrocyte, and this oligodendrocyte actually send out the cytoplasmic processes to the axons of the different neurons, and through the help of this cytoplasmic processes, these cytoplasmic processes actually wrap around the axon, and this wrapping of the cytoplasmic processes along the axon will form the myelin sheet around them. So myelin sheet is nothing but it is basically the plasma membrane of the oligodendrocyte with the cytoplasm between them. Okay, but that cytoplasm is very little because it has been removed from the plasma from between the plasma membrane. So it is literally the plasma membrane of the oligodendrocyte which forms the myelin sheath around the axon. Now, a very important point to remember here is that one oligodendrocyte actually forms a myelin sheath around many axons. Okay, then what is the importance of this myelin sheath? Actually, this myelin sheath increases the conduction velocity about 10 to 100 times, and this is actually by a conduction called as a saltatory conduction. So when we talk about the nerve impulse conduction in our upcoming videos, in that I will describe you what is saltatory conduction. But in this video, you have to just remember that oligodendrocytes form the myelin sheath in the central nervous system, and this myelin sheath insulates the axon and actually increase the conduction velocity, and that conduction process is called as saltatory conduction. Okay. Now, the another type of the cell which is responsible for the myelination in the peripheral nervous system. Okay, remember, in the peripheral nervous system is the Swan Cell. So Swan Cell actually forms a myelin sheath in the peripheral nervous system, but the oligodendrocyte form the myelin sheath in the central nervous system. Okay, so if you look at this diagram, you can see that this is one Swan Cell, and this is again another Swan Cell, or then again this is the another Swan Cell. So you can see that one Swan Cell actually forms a myelin sheath along only one axonal segment. Okay, that means that there are multiple Schwann cells which are involved in making the myelin sheath along the one axon. Okay, this is actually totally different from the scenario which is present in the central nervous system. In the central nervous system, there were one oligodendrocyte which is forming the myelination, which is forming the myelin sheath around many axons. But here you can see in the peripheral nervous system, the scenario is totally different. Here there are many Schwann cells which are making the myelin sheath around one axon. Okay, so this is because these both the spinal cord and the brain, which are the components of the central nervous system, that actually evolved inside the bony structures like the skull. Brain is developed inside the skull, and the spinal cord is developed inside the vertebrae. So that is why they are developed under very compact structures. Okay, and that is why there is a limit to the number of these cells which could be present inside these compact bony structures. Okay, and that is why the body has, you can say, the nature has developed itself in such a manner that only few cells are required for the myelination. That is why one oligodendrocyte is able to myelinate many axons. But in the case of peripheral nervous system, there is no such compact structure. So there could be one, or you can say there could be multiple Schwann cells which are involved in myelination of only one axon. Okay, so this is a major difference between the central and the peripheral nervous system. And the another thing is that Schwann cell growth factor and the debris removal by the Schwann cell and the axonal guidance function of the basal lamina are actually the factors which are responsible for the regenerative capacity of the peripheral nervous system. So this should be remembered very clearly, that the peripheral nervous system actually has the regenerative capacity, but the central nervous system doesn't have the regenerative capacity, and this is because the presence of the Schwann cell in the peripheral nervous system. So now we move on to the next type of the Neuroglia, which is the Esterocite. So Esterocite, as the name suggests, it is a star-shaped cell, and it has two forms, the protoplasmic form and the fibrous form. Protoplasmic form is found predominantly in the gray matter, and the fibrous Esterocites are found in the white matter. Okay, and now we will see the functions of the Esterocites. So Esterocites actually has a wide range of functions in the nervous system. First function of the Esterocite is that the end-fate processes of the Esterocites, they actually help in making the tight junctions between the endothelial cells, and it will effectively form the blood-brain barrier. So you know that blood-brain barrier is present outside the brain, and it actually prevents the passage of the substances between the brain and the blood. So it acts as a barrier and protects the brain. So for that, there should be tight junctions between the endothelial cells of the blood capillaries. So the tight junctions are actually ensured by the presence of the end feet of the astrocytes. Okay. The next thing is that it induces angiogenesis. Angiogenesis means that there is formation of new blood vessels. So in that way, we can say that the astrocytes actually ensure a very good blood supply to the brain tissue. It also secretes the nerve growth factors, secrete the extracellular matrix in which the neurons are embedded, and the adhesion molecules. Okay. All these things actually help in the migration of the neurons. It regulates their proliferation, differentiation, as well as the survival of the neurons, because ultimately all these things will add a nutritive value in the life of neurons. Very important function of astrocyte is a reuptake and the inactivation of the neurotransmitter. For example, in the case of glutamate. So glutamate in the synapse is taken up by the help of astrocyte, and then it will be again available for the neuron. So by this, it is basically clearing the synapse from all the neurotransmitters so that the synapse is ready for the next impulse, for the next discharge of the neurotransmitter. The next function is that it has a variety of the ion channels, like the potassium channel. So potassium is secreted during the neurotransmission. So after that, it has to be removed from the synapse, and that is why the potassium channels are present on the astrocytes, through which the potassium enters inside the astrocyte from the synapse, and this process is called as a spatial buffering. One more important function of astrocyte is the detoxification of the CNS. Is if there is any toxic metal which is present in the extracellular matrix, the astrocyte will take it up and protect the neurons from that toxic substance. Okay. So with this, we have completed the astrocytes also. Next, we move on to the Ependyma cells. So Ependyma cells are present and they line the surface of the brain ventricles. Ventricles are actually the areas, or you can say the cavities in the brain, which has the brain fluid, which has the cerebrospinal fluid, CSF, and it also lines the central canal of the spinal cord. The central canal of spinal cord also has the CSF. So apart from this, no other function of the Ependyma cells are known. So this is a diagram showing the Ependyma cells. Okay, so with this, we move on to the next type of the Neuroglia cells, that is the Microglia. So Microglia are the smallest cell in the central nervous system, and that is why it is named as micro. So it basically acts as a scavenger cell. It acts as a macrophage which actually phagocytoses all the cellular debris and clear the nervous system from any sort of debris. So it is actually derived from the macrophages which is lying outside the nervous system, and then physiologically and embryologically it is unrelated to the other types of the neural cells. Okay, so this is a diagram which is showing you that how the microglia works. Okay, so when there is no infection in the nervous system, when there is everything perfect, the microglia are basically in the ramified stage. Ramified stage means that there are so many cytoplasmic processes which is extending from the body of this microglia. Okay, but once in response to the pathological states, the microglia gets activated and it's get converted into the reactive microglia. Now the reactive microglia actually has a different morphology as compared to the resting microglia. So now here you can see there is absence of the branching pattern, and there will be presence of lysosomes and phagosomes. Okay, so this microglia, this reactive microglia, will now called as an active brain macrophage, will actually phagocytose this degenerating cell or the degenerating neuron, and then it will undergo the process of apoptosis, that is a programmed cell death. Okay, so this is basically happen when there is immune system activation in the central nervous system. Infection came, and in response to the infection there is change in the morphology of the microglia cells, and it will phagocytose that infectious agent and remove it by the help of programmed cell death. Okay, So with this we reach at the end of this video. Please like, share and subscribe our channel. Please let us know any topic you want us to make a video, because it will really help me to decide that what should I post next on my channel. You can follow HM Learnings on Facebook, Instagram and Twitter. And as you know, we have started sharing our contents on website SlideShare. I actually share the SlideShare link of every video in the description box, so you don't need to go anywhere. You just click on that link and you will be on the SlideShare. And if you want me to teach you personally, you can contact me on the email ID I shared on the description box, or you can direct message me on any of my official pages. Okay, till then, keep learning.